If I started with a survey like we did yesterday, what is Abivax about? If I started now, you would probably say, oh, vaccine company. I hope at the end, if we did the same survey, you would remember that Abivax, unfortunately, is not a vaccine company. So it's a misnomer. Abivax was created 5 years ago. A very small company, 24 people, and at that time, yes, we had a vaccine program. Abivax was formed out of 3 small companies, which were formed by Philippe Pouletti, immunologist and entrepreneur. And one of these companies had a licensing with a therapeutic Hep B vaccine, which unfortunately, in the meantime, Abivax discontinued. But the name remained, so we are not a vaccine company anymore. So hopefully, if we did a survey at the end, you will remember that. So today, what I wanted to go through is a 464. Which, from its formula, you can see is of the family of the quinolines. And yeah, these are my disclosures. I worked 30 years for GSK and ViiV, so it's normal I put that up as well. So it's a small molecule. It's an oral molecule, which actually has been developed initially by, as I said, one of the previous companies leading to Abivax, which was called Splikos. And Splikos was generated at the CNRS in Montpellier by Jamal Tazi, Professor Jamal Tazi, who is in the audience here, who has spent, I think, his life around RNA biogenesis and splicing. And at some point in time realized that pharmacological agents could interfere with this process. And we all know that in order to have HIV replication, as you can see on the left-hand side of this slide, you have a ref which is there, which prevents the splicing from going ahead and allowing for these large structural proteins to be formed and replication to happen. What happened in the event of when ABX464 is being added is that it interferes with this process, and actually the splicing does not go ahead anymore. There's no replication. Now, this explains very nicely why this drug could be another antiretroviral. And I can tell you, I spent 30 years in large pharma developing antiretrovirals. I did not join this company to develop another antiretroviral because I think the market is well served, despite the fact that we may need new modes of action. But what was really interesting was when they put this drug into the humanized mouse model, is that the beauty of this model is, of course, is that these mice respond like humans. If you infect them, you see the viral load going up 10 to the 4th, 100,000 copies. And the viral load goes down if you treat these mice with triple therapy to undetectable. And of course, as we all know, like in humans, 10, 14 days after stopping treatment, the viral load comes back up to pre-treatment values. Now, when they compared it with 464, they saw that, of course, because it's monotherapy, the viral load does not come down at the same speed and at the same depth. But the big surprise came when the drug was stopped, that the viral load did not come back to pre-treatment values. It remained, instead of going back to 100,000 copies, it remained at about 4,000 copies. And that was 2 weeks after stopping the treatments and 6 weeks after stopping the treatment. And these are the data, I must admit, which convinced me, well, maybe there's something else there. Because this cannot be explained by the drug itself, which has a half-life of a couple of hours. The main metabolite and glucuronide has a half-life of a couple of days. So this cannot explain why 2 weeks after stopping this treatment, you still had no new virus being formed. You have the viral load remaining low at 4,000 copies and also at 6 weeks later. So, in terms of mechanisms of action, as I said, the left part here is easy to explain in terms of RNA splicing. Yes, there's splicing going ahead, and therefore there's no viral structural proteins being formed. So, in terms of reduction of viral load, that's easy to explain. On the right-hand side, you also see that the drug actually, and I will not go into detail there, but the drug has anti-inflammatory properties. And through a long time, unfortunately, we're not allowed to go through that, but we are now developing the drug also in ulcerative colitis in phase 2. because the drug was shown in the DSS model that actually it has very potent anti-inflammatory properties. Now, inflammation is not that far from reservoir, as we all know. And of course, what we have to elucidate now is how the reservoir is being affected by this drug. And for that, there are 3 hypotheses which are being investigated as we speak. But the nice thing about it, because initially we thought, okay, you have a drug which does this, which inhibits viral replication. You have a drug which does, which we have shown, and I will show you later, which impacts the reservoir. And you have a drug which also, in a total different indication, has an effect on inflammation. How do you put that together? And I think that's the beauty of the poster which is at this meeting, showing that actually there's one common molecular target, which is cap binding complex 8020. And I think Jamal's data, which we can send you the poster, just send us an email, info@abivax.com, and there will be, of course, we are submitting now to paper, the paper to being published, but that really the CBC 8020 complex is the cornerstone of explaining these different actions, which may be of importance, of course, for ulcerative colitis, that it's not an off-target effect, as some people were fearing, and we as well, that we said, well, how it comes that drug does something totally different there, without the presence of a virus, of course. But also that the inflammation, as we know, is very important in HIV, that this may have an additional benefit for patients to that. And that's all going through thanks to the increased expression of this anti-inflammatory MIR-124. And that was, I think, a very important progress which we made in terms of mechanism of action of ABX464. So far we have given this drug, I'll come back now to the HIV program, to more than 180 patients and healthy volunteers. In terms of safety, I'll be very brief, but we'll go in more detail later, is that actually it has some, at the highest dose, it has some GI symptoms, nausea, vomiting, and some headache initially. It's a bit like when you give your triple therapy to patients, the first couple of days they might have this. We see that as well here. It has antiviral properties. We showed that at the CROI 2 years ago, that the drug indeed in treatment-naive patients has antiviral properties. We showed now that in 2 independent studies that HIV DNA could be reduced and that it's feasible, and also that the single copy assay showed a reduction in case there was a residual viremia. And that MIR-124 is being overexpressed in tissue, and we are planning the phase 2b study. The first study which showed that indeed this drug could have an impact on the reservoir was the 004 study, which was presented at EACS last year, where we gave the drug on top of boosted darinavirine, patients who were well suppressed on boosted darinavirine. You may ask, well, why boosted darinavirine? Well, we are a small biotech. At that time, we didn't have drug-drug interaction studies with other drugs, so we wanted to limit it to what we had. Why 28 days? Again, we're a biotech company. We did have the data at that time which allowed us to go for treatment for 28 days in terms of tox. Today we can go for 1 year and more. Ulcerative colitis has now been approved for its clinical program to be treated by over 1 year. But at that time that was the position we were in. And so we did that, and we added 464 versus placebo to the patients. And what we saw was that actually quite surprising, because I had asked to the investigators, which were amongst them in the room here in Spain, Belgium, and France, they asked them what their expectations were. And they said, well, if we see a 20% reduction in 20% of the patients, it would be already not bad. And what we saw was in 53% of the patients, a 36% reduction. So it was a first signal. I'm very careful, and you will not hear me saying, oh, we have a cure here. Not at all. I mean, this is a first signal, but an important signal that indeed it is achievable to get to a viral total HIV DNA reduction. So the second study, which we embarked on in parallel, was to look at not only the compartment of the blood, PBMCs in the blood, the total HIV DNA, but also in a different compartment, i.e. in the gut, and to see whether through rectal biopsies we could also see a decrease in total HIV DNA in that compartment. And that study actually again was with 150 milligram for 28 days for the same reasons to patients who were under triple therapy this time. And this study is being performed in Barcelona. And we are doing sampling, of course, blood sampling, but also rectal tissue. And these are the data on baseline demographics. Safety, there were 2 withdrawals. Again, as I said, at 150 milligram, which is the highest dose, you can have abdominal pain, some headache, but all discontinued when the patient stopped the treatment. Important. This was the second study where we showed, well, actually, Javier Martinez-Picado showed that you could see a decrease in total PBMCs, sorry, in HIV, total HIV DNA in CD4 positive T cells between day 0 and day 28 by up to 50%. You see a variability, but don't forget, this is only 28 days of treatment in addition to triple therapy. As I said, those patients who had residual plasma viremia, we saw that in all of them we saw a reduction in it during the treatment period. The biopsies were unfortunately not of good quality in order to look at total HIV DNA. However, we had frozen biopsies which allowed us to look at the anti-inflammatory markers. And we saw that miR-124, which we believe is explaining why this drug has anti-inflammatory properties, was increased in all patients overexpressed in all patients between day 0 and day 28, and went down when the drug had been stopped. So which showed there's a pharmacodynamic effect of our drug, and the drug really works at that level as well. So I will not go through these conclusions, but rather to the next steps, because I think that's more important. The second cohort of that study is now looking at not 28 days, but 84 days. So 3 months of treatment, how can we see a further decrease in total HIV DNA? And moreover, can we also see it in the rectal tissue, which this time we are absolutely sure that we're going to have good quality cells in order to assess that. We should have this data by July of this year, and that will, of course, allow us then to go to phase 2B. The phase 2B study, which is being planned as we speak, is really looking at whether treating patients with 464 over up to 1 year, how long it takes for each patient to get to the lowest level possible total HIV DNA. And only when these patients have reached that low level, then can we propose a treatment interruption and potentially look for an exploratory way whether indeed we have an extension of time to viral load rebound. Because of course, if you reduce the reservoir as we saw it as measured by total HIV DNA by 50%, you still have 50% left. And no surprise that if you do a treatment interruption at that time, your viral load will come back immediately within 10 to 14 days. So we hope that by treating longer, so up to 1 year, in addition to triple therapy, that we will be able to achieve a reduction in total HIV DNA, which would allow at that time to look for treatment interruption. This study is now being planned, and the protocol is being finalized. Of course, there will be consultation with community and patient organizations on that. I must admit I'm thrilled about the support I have from a number of you here in this audience, but also in the US, who want to participate. Some centers say, we can do it on our own because this is easy to do, right? We don't, from experience, I know we have we shouldn't put all our eggs in one basket, but I think the enthusiasm from the scientific community actually is great to have all these people wanting to participate to that. So what we should get from this phase 2B study is really, indeed, as I said, can we achieve a lowest level possible of total HIV DNA? And that's a big challenge for us, of course, total HIV DNA being an academic lab assay. Now we're moving towards, if you want to go into phase 2B, 180 patients, you have to go through a GLP accredited assay. So of course it's a hurdle. And we're going to look at two, because we're going to stratify initially the patients in high and low total HIV DNA at start and see whether there's an impact of that. We're also going to look at two doses, whether 50 milligram or 100 milligram makes a difference. We're also going to have another study looking at the early treated patients. This is a cohort in the UK where these patients were treated early, and these are the only patients where we know that they have a low reservoir at start. So, as Jonathan said yesterday, inflammatory markers are important, but which ones? And I think that's a big challenge for us as well to know, to see what we can see there. Of course, the holy grail is whether we can go for treatment interruption and to see how many patients after treatment of up to 1 year with this drug, in addition to triple therapy, would be eligible, and what the outcome would be in terms of treatment interruption. So I will stop here, and I really want to thank. There are many in this room who contributed and who are extremely enthusiastic about this program. I really wanted to thank you for that and take any questions you have. Thank you.